Hello dear students, welcome back to another learning lessons in my Physio-Pulse channels. Today we will discuss about the protein metabolism in exam-oriented way and in a simple, easy to understand way. You will find the clear explanations, diagram and important concepts here. So before begin if you find this video helpful, please like, share and subscribe to support the channel. Now let's understand about the protein metabolism. Actually, protein metabolism involves in deamination and transaminations. What is the meaning of deamination? Deamination is the removal of the amine group from the alpha amino acids as a ammonia with the formation of alpha keto acids. That means amino acids will be converted to form the keto acids. And the liver and kidney are the main sites of this deamination process. So sites of this deamination is liver and kidney. And it may be oxidative or also it may be non-oxidative. So according to this, it is classified into two ways. One is oxidative deaminations, another is non-oxidative deaminations. First of all, oxidative deamination is catalyzed by one of the following enzymes, that is alpha amino acids oxidase. This enzyme is present in the liver and kidney, and it is its activity is very low. It is an aerobic dehydrogenase that needs FMN as the coenzyme, and it deaminates most of the naturally occurring alpha amino acids. So it is an example: alpha amino acids FMN, then alpha amino acid oxidase. It will convert it to alpha amino acids, then it is alpha keto acids. So in this way, alpha amino oxidase, it helps for amino acids convert to the keto acids. Secondly, D-amino acids oxidase. This amino acids, D-amino acids are present in the plants and bacterial cell wall, and most of them are not used in protein synthesis in human as well as the animals. Now to see how D-amino acids are deaminated by the D-amino acid oxidase, resulting to the ammonia and keto acids. So it is this type of D-amino acids are present in the liver, and it is aerobically dehydrogenase and needs the FMN as a coenzyme. So D-amino acids to amino acids, then keto acids. The enzyme is D-amino acids oxidase. And number three, glutamate dehydrogenase. This enzyme is also present in the cytoplasm, mitochondria, and the most tissues. Its activity is high, and it is anaerobically it is dehydrogenase, and it needs NAD and NADP as a coenzyme. It determines the glutamic acids and also resulting alpha ketoglutarate acids and ammonia. So this is the example: glutamic acids, it convert to form the alpha aminoglutaric acids, then alpha ketoglutarate acids with the help of glutamate dehydrogenase. So we have already discussed the oxidative deaminations. Now, what is non-oxidative deaminations? That is, it catalyzes by one of the following enzymes, that is dehydratates. This enzyme deaminates amino acids containing the hydroxyl group, that is serine residue or homoserine or threonine. It needs pyridoxal phosphate as a coenzyme. So example is given here. This is serine dehydratase enzyme, alpha amino acids and pyruvic acids. And number two is desulfidase. This enzyme deaminates sulfur containing amino acids, that is the cysteine and cysteine. It needs the pyridoxal phosphate as a coenzyme. So cysteine can be converted to alpha amino acids and pyruvic acids. So in this way, deaminations we can discuss, or we understand that deaminations process, that is the oxidative and as well as the non-oxidative pathway or non-oxidative deaminations. Now, what do you mean by transaminations? So students, you will find that transaminations mean the transfer of amine group from an alpha amino acids to form the alpha keto acids. So thus creating a new alpha amino acids and a alpha keto acids. So this pathway is responsible for the deamination of the most amino acids, and this is the most important major degradation pathway which converts the essential amino acids into the non-essential amino acids. So this pathway amino acids that can be synthesized by de novo D pathway by the organisms. And the liver is the main site of the transaminations, and all the amino acids can be transaminated except the lysine, threonine, proline, hydroxyproline. All transamination reactions are reversible, and it is catalyzed by the aminotransferase, that is the transaminase, and it needs the pyridoxal phosphates as a coenzyme. So in this way, the figure we are explaining here, this is the how it is happened. Now, role of pyridoxal phosphate in transamination. What is the importance of the pyridoxal phosphates? This pyridoxal phosphates acts as an intermediate carrier for amino group, and it accepts the amino group from the amino acids to form the pyridoxal phosphate, which in turn gives the amino acid to the keto acid, alpha keto acids. So these alpha amino acids will be converted to form the alpha keto acids. This pyridoxal phosphate acts as a carrier of cofactors intermediate. And then these alpha amino acids will form the new alpha keto acids. For example, if you see the alanine transaminase, aspartate transaminase, glutamate transaminase, then all these enzymes can help in this way transaminated. Now, alanine transaminase, this is very important because of that. It is called the also called the GPT, that means glutamic pyruvic transaminase, and also catalyze the reverse reaction and catalyze the transfer of amino group from glutamic acid to the pyruvic acid to form the alanine and alpha ketoglutamate acids. It is present in the cytoplasm of the liver. So this is the figure which we are explaining here, pyruvic acid and to form the alanine, and then this is alpha ketoglutamate acids and glutamic acids. Now second is the aspartate transaminase. If you see the aspartate transaminase, it is also one of the important SGPT and SGOT. We have already found in our lecture of enzymes. So it is also called the glutamic oxaloacetic acid transaminase. Also catalyzes the reverse reaction. It catalyzes the transfer of amino group from the glutamic acid to the oxaloacetic acid to form the aspartic acid and alpha glutaric acid, alpha ketoglutamate acids. So this is the example of this alpha keto to alpha ketoglutamate acids, how it is formed and from glutamic acids. Now, what are the clinical significance of this serum transaminase? Well, actually, you see that transaminase are intracellular enzymes. Their levels in the blood plasma are low under the normal conditions. So, any damage to the organ associated with these enzymes, like liver or heart, skeletal muscles, will increase the level of transaminase in the blood. So, in liver disease, there is an increase both serum ALT, that is SGPT, and SGOT level. So, doctors can prescribe to see or to review their SGPT level or SGOT level when the liver is not functioning properly. During the acute liver disease, that is viral hepatitis, the increase is more in SGPT. In chronic liver disease, the increase is more in SGOT. And in heart disease, myocardial infarction, skeletal muscle disease, myasthenia gravis, also it is increases in SGOT level. Now to see the difference between transamination and deamination. Actually, if you see the process of transamination, the transfer of amino group from one molecule to another group, and especially from an amino acid to the alpha keto acids, and in case of deamination, the removal of amino group from an amino acid to the alpha keto acids. And now the process involves in the synthesis of non-essential amino acids, whereas the process involves in the breakdown of excess proteins. Where it occurs specifically in the liver cells, heart muscles, and skeletal muscles. And it occurs in the liver and kidney cells. What are the enzymes? Exactly, in case of transamination, transaminase and amino transferase catalyze these reactions, whereas oxidase, dehydrogenase, dehydratase, all these are acting as the respective enzyme for the deaminations. And the result is an exchange of alpha amino group to the alpha keto group, and the result here the elimination of ammonia. Now, specialty, that the glutamic acid is the main form of amino acids produced in this reaction, and glutamic acid is the primary form of amino acids which undergo the deamination. And the deamination is transamination is the reversible, while the deamination is the irreversible. And associated enzyme pyridoxal phosphates, we have the FMN, FAD, NAD+. Non-oxidative pyridoxal phosphates are actually associated enzyme. So in this way, we can differentiate between transamination and deamination, and which is very, very important for your exam purposes. So students, we already discussed or covered our lectures with the transamination and different types of transamination and deamination processes, how it occurs and what are the enzymes or example of the transamination processes, and even also non-oxidative pathway of deamination and unoxidative pathway of deamination we have elaborately discussed here. So if you found this lecture helpful, do not forget to like, share, and subscribe to the channel. Press the notification bell so you never miss out our next upcoming biology, biochemistry, physiology lectures. Thank you for watching, and we'll see you in our next lectures. That is